In this module, we will discuss and study the transcriptional mapping, the mapping of the genes on the sites, which is a very, very important step toward understanding the gene, the gene sites, gene action, and the gene function in the cell. Now, the study which deals with the transcriptional mapping is known as the transcriptomics studies. Now, transcripts, they can be mapped with great accuracy to sites in the whole chromosome, that where in the chromosome they are situated. And this kind of study, called the transcriptional mapping, helps to understand the different functions, placement, and the control of those genes. Now, transcripts of the protein encoding genes, for example, those genes which encode the protein, they make up only a small fraction of the whole human transcriptome. जो whole human transcriptome जो है उसका छोटा सा हिस्सा जो है ये प्रोटीन इनकोडिंग जीन्स जो हैं वो बनाते हैं. If we consider only exons in protein coding genes, we would predict that only 1 to 2% of the whole human genome would be expressed in RNAs, ribonucleic acids, which is found in the cytoplasm of the cell. Remember, only 1 to 2% of the whole human genome, a small quantity. Transcripts of unknown functions are also there. And the excess of unexpected transcripts has been dubbed transcripts of unknown function, that is TUFs, transcripts of unknown function. Definitely, there are those transcripts which we still presently don't know what their function is, but they are there. These are tufts. Now, transcribed fragments. What are the transcribed fragments? Now, all of the transcribed regions, exon and tufts alike, they are detected by such arrays and are called transcribed fragments, or the transfrags. That is, those areas, those fragments which are on the transcribed regions and where they are exons and the tufts, which are detected by such arrays, they are called transcribed fragments, or the transfrags. Approximately 2/3 of the transcripts in human cells have been reported to be non-polyadenylated A. And it means that the poly-A, non-polyadenylated poly-A, that is the 2/3 of these are known as the non-polyadenylated fragments. That's the poly-A. Now, in the figure, the proportion of each of the 10 human chromosomes from which cytoplasmic polyadenylated transcripts are made is shown. And such transcripts from intergenic regions and introns are, by definition, unannotated. They are not definitely known, they are not labeled, and as we see in this, you can see that the transcription map of 10 human chromosomes and the tufts which are there and how much that is formed in the chromosomes. Now, these regions make up the majority, that is the 57% of the transcripts from the 10 chromosomes as a whole. Kitne? How much? 57% of the transcript from the 10 chromosomes, they are made by these regions. And studies which show abundant cytoplasmic poly A plus and poly A negative transcripts of non-exon regions, they may help to explain the differences between the organism. The advantage is that the studies which show abundant cytoplasmic poly A plus and the poly A negative transcripts from the non-exon region, they can explain the differences between the organism, the differences existing between different organisms can be explained by this. Although the exons of human and chimpanzees are extremely similar, the non-exon regions, they have diverged considerably more. So that is how we can find out that how they differentiate between different organisms and what the advantage of the chromosome transcriptional mapping is exactly this.